Okay, everybody. So I think we're going right now to our last presentation of this session. You have 20 minutes for presenting. Please keep an eye on the clock. And we're gonna have 5 minutes afterwards for the questions of the judges. If we have more time, we can also have questions from the audience. This is say hi to the team of Bielefeld-CeBiTec. Yeah.
Hello, my name is Leonard. I'm Chris, and we are members of the iGEM team Bielefeld-CeBiTec. Welcome to our presentation expanding to genetic code. With this project, we want to open a complete new field of synthetic biology for the iGEM community. First, I want to start talking about proteins. Proteins are the molecular machines that perform chemistry in life. Through protein design and engineering, we can generate new proteins with interesting applications for industry, medicine, diagnostic, and basic research. But the properties of those proteins are limited to the properties of the naturally occurring canonical amino acids. Therefore, we want to incorporate non-canonical amino acids with new exciting properties for the proteins. With non-canonical amino acids, you can develop new antibiotics against multi-resistant bacteria, or you can enable post-translational modifications for protein expression in E. coli. So, therefore, we offer new possibilities for advanced protein design and engineering. So, how do we incorporate amino acids in general? We need a tRNA that is specific for the amino acid and for the codon. And then the amino acid gets incorporated by the translation machinery into the polypeptide. So how do we incorporate non-canonical amino acids? Therefore, we need to reallocate an existing codon like the AMBER stop codon. And therefore, we need a tRNA that is specific for the reallocated codon and for the non-canonical amino acid. And this requires a new amino acid synthetase that recognizes a non-canonical amino acid and loads it to the tRNA. So how do we obtain new tRNA synthetases? We can either model new tRNA synthetases in silico, or we can evolve new tRNA synthetases by selecting a tRNA synthetase library. And this is how we obtained adapted synthetases. We developed a tRNA synthetase library by randomizing 3 amino acids inside the binding site of the tyrosine tRNA synthetase, and we were analyzing this by Illumina sequencing, and this was performed by our team member Laura. And the results showed that we had a high diversity for our library, covering over 95% of all possible peptide sequences for the tRNA synthetase. So now that we have the library, we need a selection system for the incorporation of non-canonical amino acids. And therefore, we contacted a leading expert for tRNA synthetases, Professor Brieser from TU Berlin, and he gave us a chance to have one of our team members working at this laboratory in Berlin and working on selection system. And we developed a 2-plasmid selection system, and we performed a selection for the incorporation of the non-canonical amino acid nitrophenylalanine. So we first performed the positive selection for functionality, and then went into a negative selection for the specificity. So in the end, we obtained about 100 colonies, each containing a tRNA synthetase, a functional synthetase that is specific for nitrophenylalanine. A parallel approach we did was the in silico modeling. It's a structure-based modeling. So therefore, we had to model a tRNA synthetase for a complete new non-canonical amino acid, CBT asparagine. And we used the software Rosetta. Rosetta was simulating mutations inside the binding site of the tRNA synthetase. And for feedback, we contacted Florian Richter, who is the developer of Rosetta protocol, for enzyme design. And regarding this feedback, we optimized our modeling, and we obtained new peptide sequences for tRNA synthetases. And going back to the wet lab, we were able to validate our tRNA synthetases with our selection system in vivo. So therefore, we are the first iGEM team that implemented a Rosetta Protocol workflow and fully documented on the wiki. And we were able to construct a library that is available for the iGEM community on request, and we can submit a library. And we have a selection system for the TNR synthetases. So what did I show you? We demonstrated how to model new TNR synthetases either in silico or by evolving them, selecting them through a library. And now I want to come to new, exciting, non-canonical amino acids, and therefore we developed a toolbox for 5 applications for our non-canonical amino acids. And these are the 5 applications. The first, analyzing, there's photo switching, there's labeling, we have photolysis, and fusing. And here I want to present 2 of our applications. The first one is photolysis. Photolysis can be performed by the non-canonical amino acid nitrophenylalanine. So incorporated into the polypeptide, it can cleave the backbone of the polypeptide by simply light irradiation with UV light. And we thought that this could be a really cool application for a new method for protein purification. And we are giving the molecular base. So we have the target protein that is linked to an affinity tag, and this linker is containing the nitrophenylalanine. So we can immobilize it on a column, then we would perform the elution with UV light, and showing in this Western blot, the irradiation of the UV light is cleaving off the target protein from the affinity tag. So yes. And we talked to 2 co-founders of a biotech company, and they said this could be industrially relevant for fragile proteins since it's a mild elution method. So fragile proteins like the blood clotting protein factor 8. Therefore, we created a new method for protein purification by light-induced cleavage. The second application is fusing, and therefore we have to design and synthesize a complete new non-canonical amino acid, and this is CBT asparagine. This synthesis was performed by Daniel, and this non-canonical amino acid can fuse with a covalent bond to the side chain of a second non-canonical amino acid, is cysteine lysine. And this fusing is performed under physiological conditions, and we proved this by mass spectroscopy. So therefore, we can fuse 2 polypeptide chains, terminus independent, with this non-canonical amino acid. We thank Sandeep Jadhav for supporting us during the synthesis. He's from the Department of Organic Chemistry. So we were able to synthesize a novel non-canonical amino acid. And this shows that synthetic biology is not limited to just the naturally occurring non-canonical amino acid, but one can think of his own non-canonical amino acid and synthesizes and uses for a specific application. And we also modeled the TNR synthetase with our Rosetta protocol for this new non-canonical amino acid. Since we had to deal a lot with light irradiation for our toolbox, we developed a new hardware. This is the LED panel. With this LED panel, you can irradiate the 96 well plate with either UV light or blue light. And it went through several cycles of design, build, and testing, and so was a lot of prototyping. We also 3D printed a case for easy handling. And to rebuild this, the menu or the constructions to rebuild this can be found on our wiki. We also developed a new Android application that can be used for an easy control via Bluetooth. And we used it to validate our toolbox. We were able to do irradiation experiments like photo switching and photolysis, and this shows that this LED panel can be easily adapted to any experiment that requires some kind of irradiation. So we developed an adjustable LED panel and check it out at the exhibition space at Table A for further information. So I showed you how to evolve new TNR synthetases for the incorporation of non-canonical amino acid, either by in silico modeling or by the selection of the library. And I presented a toolbox, and we demonstrated that we incorporated non-canonical amino acids and showed the applications.
Yeah, but with this approach, we have to face certain limitations, and that is that we can only incorporate one non-canonical amino acid. If we also reprogram, for example, a lysine codon, we can incorporate two, but still we are very limited in the number and in the flexibility. And additionally, reprogramming existing codons leads to loss of function of host cell proteins, since these codons also occur somewhere else in the cell. So therefore, we want to come up with a new solution, and that is introducing unnatural base pairs. By adding just one new nucleobase, we can create 61 new blank codons that are not used by the cell in any other way. And if we introduce an entirely new base pair, we have 152 codons that we can use to incorporate non-canonical amino acids. So for our project, we use the unnatural base pair formed between iso G and iso C, in our case a methylated form of iso C. And to create such a semi-synthetic organism with a new base pair, we have to face certain challenges. And that is that we have to somehow facilitate uptake of these new nucleotides, or we have to produce them by biosynthesis. We have to develop or somehow make sure that we can retain them in the DNA, and we have to develop new methods for detection as well as basic molecular biology. And of course, since it's a very new field, we also have to deal with social and scientific implications. That's what I want to start with. So for our project, we wrote the CHIMP report, which stands for Chances and Implications of an Expanded Genetic Code. And for that report, we interviewed several experts from different fields, including ethics, philosophy, social studies, scientists, as well as religious representatives, including, for example, Professor Alta Chowra, former bioethicist for President Obama. The results from these interviews then went into our framework, which we developed to ensure safe and responsible research on expanded genetic code. So therefore, we dealt with the ethical, philosophical, scientific, and social implications, and developed a framework that other teams can use to ensure safe and responsible research on an expanded genetic code. So how do we take up unnatural nucleotides? Therefore, we used an algae transporter, a nucleotide transporter called PTNT2 from the algae Phaeodactylum trichonucleum. And how it takes up these nucleotides is that it takes them up in exchange for ATP. So cells that take up these unnatural nucleotides export ATP, which is then converted to AMP. So based on signal peptide predictions and topology predictions, we designed and tested 5 different variants to find the best suited one that offers the best growth and uptake compromise. So we were able to study the subcellular localization by isolating the inner membrane fraction, performing Western blots. We're also able to show that the transporter is located in the membrane with laser scanning microscopy with GFP-TEC variants. And to study the function, we perform cultivations. We're able to show that different transporter variants have substantially different growth characteristics. We're also able to show that the AMP levels in the supernatant of strains expressing our transporter are higher, therefore they export ATP, and we're also able to test the uptake efficiency of our transporters. And based on that, we were able to find the best candidate that offers the best compromise between growth and uptake. So therefore, we successfully demonstrated uptake of our unnatural nucleotides iso-DCMTP and iso-DGTP, and characterized all of these transporter variants and handed them in as biobricks. But during our research, we also came across a paper from the 1930s about a plant called Croton tibium. And that plant is also used in traditional Chinese medicine, and it was reported that it produces one of our natural bases. So that kind of was a cool thing. So, but we found out there was nothing published on it, no genome, no pathways whatsoever. So we went to a botanical garden and collected some samples from the plant, performed RNA isolation, RNA-seq, assembled a transcriptome, performed annotation, and then searched for possible gene candidates that might be involved in the synthesis of this new base, or unnatural base. And we found one that we cloned in E. coli, expressed and purified, and were then able to study the enzyme activity. And in the end, we were able to show that the gene that we identified, the GMPS isoform 2, so a new isoform of the guanosine monophosphate synthase, actually produces a product that differs from GMP. So therefore, we concluded this is an enzyme that produces one of our unnatural bases. We were the very first, to the best of our knowledge, to do so with enzymatic synthesis. And we also added this to the partner registry so that other teams can build on this and produce these unnatural bases themselves. How do we actually retain an unnatural base once we have it in the DNA, which we do via gene synthesis? Therefore, we use a CRISPR-Cas9 based retention system. So how it works is pretty easy. We have 5 different single-guide RNAs that cover all possible mutations. If a mutation occurs, one of these single-guide RNAs can bind, and therefore Cas9 can cut the plasmid, which is then digested by nucleases. We included this in our two plasmid retention system. One plasmid is a high copy plasmid that contains Cas9 in our transporter. The other one is a low copy plasmid which carries the single-guide RNAs and MRFP fusion protein, MRFP and SACP fusion protein, in between which is the insertion site for our unnatural base pair. So for that, we worked with an expert from London. He gave us very valuable advice regarding the toxicity of our natural base pairs on E. coli cells. So therefore, we developed a two plasmid-based retention system, and in one experiment, we were also able to show that unnatural base pairs are still present after 48 hours in vivo, but these were preliminary results, and further testing needs to be performed for precise quantification. But how do we actually detect unnatural base pairs? And that's what our software called iGEM comes in. iGEM is a software that adapts Oxford nanopore sequencing for the detection of unnatural base pairs. So how it works is a DNA strand is pulled through a pore, and as the DNA goes through the pore, the current over the membrane changes. We can then analyze and interpret the signal and the change to identify which pore is currently inside of the, which base is currently inside of the pore. We did that, and we're able to show that the signals from the natural bases, so the top four, differ from the signal that we get from our unnatural base. We then performed linear discriminant analysis to show that the signals obtained can clearly be distinguished from one another, which means that we can perform highly precise predictions on which base is currently inside of the pore. So out of that, we performed further validation with more experimental data, and all of that is automatically done by our software, which means we successfully adapted Oxford nanopore sequencing for the detection of unnatural base pairs. And as far as we know, we are the very first to ever do so with this sequencing technology. But given that Oxford nanopore sequencing is rather expensive, and not every iGEM team has access to it, we wanted to come up with a new solution that is more kind of in the iGEM spirit, that everyone can use it. And that's where our second software called MAX comes in. MAX stands for Mutational Analysis Explorer. And it's a database search tool that searches restriction enzyme databases for restriction enzymes that have overlapping recognition sequences. And they all differ in only one position, which is where the unnatural base pair sits. So if the unnatural base pair is present, it efficiently blocks off the recognition of these enzymes. And therefore, we can not only say if a mutation occurs, but also which one. So in this case, it's a real experiment we performed. A mutation to A occurred, and therefore the enzyme ECI1 was able to cut. And therefore, we can say, okay, that a mutation to A occurred. We were also able to validate this with our adapted Oxford nanopore sequencing protocol, and we're able to show that this is an orthogonal method that leads to the same results. So therefore, we developed an inexpensive and easy-to-use method for the detection of unnatural base pairs that every iGEM team can use, because all you need are 4 enzymes. So therefore, we successfully demonstrated the uptake of our unnatural nucleotides. We identified the gene in the plant called proton-TPM L as a producer of our unnatural base, iGEM. We developed a retention system. We're also able to develop 2 new methods for the detection of unnatural base pairs, and also dealt with the social and scientific implications of our new approach. So therefore, we successfully demonstrated the translation and incorporation of non-canonical amino acids, and therefore expanding the genetic code with new amino acids. And we also introduced unnatural bases to the iGEM community, and therefore expanded the genetic code with new bases, which we hope further future teams will build on to then eventually incorporate non-canonical amino acids with non-canonical codons. And with that, we would like to thank all of our supervisors and advisors, the teams that we collaborated with, CEO Boulder sitting right here, the first or only iGEM team from Italy this year, UNIFI, TU Darmstadt, and of course all the research groups that were involved, all of our sponsors, partners, and of course all of you for being here. Thank you.
Erst das Team durchlassen.
Okay, thank you. Now we're going to pass to 5 minutes for the questions of the judges.
Yeah, really interesting work. Could you talk a little bit more? So, there's growing interest, obviously, in this idea of using non-canonical amino acids for protein production. One of the biggest challenges is the loss of specificity for the incorporation of residues into proteins. Did you do any assessment of that in terms of the, obviously, the work you're doing? Did you look at the fidelity of the protein sequences you're producing by incorporating these non-canonical amino acids?
Yes, actually. Actually, for our applications, we were able to show that the amino acids we want to use were functional. So we validate the incorporation rate through SES pages. So we supplemented the non-canonical amino acid into the media, and for another experiment we didn't. And we were able to show that even if the non-canonical amino acid isn't in the media, our protein of interest is expressed. So there is unspecific incorporation of canonical amino acids. That's one of the reasons why we actually did the modeling, because if there are more mutations in our synthase, it gets more specific and more orthogonal to the naturally occurring synthases in E. coli or the organisms we want to use for getting synthase of.
Yeah, I guess specifically, at what frequency do you see misincorporation of a particular amino acid where you'd expect to see one residue? And did you do any assessment of that? Because that seems to have been that's one of the major issues with currently the research work that's going on in this field is the frequency at which you get incorporation of the wrong residue.
Actually, we weren't able to show that completely because the incorporation of wrong amino acid depends on if the non-canonical amino acid is in the media. So if the non-canonical amino acid is in the media, it is incorporated more often than not the canonical, but if it isn't in the media, it is incorporated non-canonical amino acids more often. So the affinity of the synthase is higher for this non-canonical amino acid we want to incorporate than to the canonical.
So there are several ways to prove if the non-canonical amino acid is incorporated or not. One nice way is the function of the non-canonical amino acid itself. So, for example, we had a fluorescent non-canonical amino acid, and as Svenja said, we did an experiment where we produced a protein with and without the non-canonical amino acid. And on the SDS pages, we had nearly the same amount of protein, but only one of the proteins really showed this fluorescence because the fluorescent amino acid was incorporated. We also worked on a part that was, I think, done by Texas 2014 and improved it, which was a validation system for the synthases. It's a fusion protein where we have two fluorescent proteins at the linker with a stop codon, and then you can cultivate it with and without the amino acid, and then you can check if there are any differences in the fluorescent spectra. And so we could show for even some of our synthases that there are differences in the efficiency and the specificity, and that you have always the possibility to improve synthesases through modeling or through additional steps in the selection system.
I have another question if no one else. Could you say a little bit more, I liked your illuminator system for the 96-well plate. Could you say a little bit more about what the advantages of that particular approach would be? For example, it seemed like you were essentially producing uniform illumination. Was that specifically the purpose?
Yes, that was the purpose. So, we had access to a plate reader, for example, where we could illuminate our samples with a specific wavelength we wanted. But the disadvantage of those systems is that they are very expensive, and you don't want to illuminate a sample over several hours with high light intensity, if it is even possible to reach the light intensities that are intended for the experiment. And therefore, we quickly decided to create our own device, and we designed it in a way that all the wells are evenly illuminated, and you can even adjust the intensity for every well individually. And therefore, you can create or conduct several experiments in parallel, like 96 in parallel, and with a device that costs less than $200, and even much less if it is not produced in a low scale, but in a higher scale. So, and by this PCB design that we decided for, it is even much easier for other iGEM teams to rebuild this, or for teams in general, because you simply have to go to your board manufacturer and order such a board, which is not which is quite cost efficient, and you cannot do something wrong. All the circuitry is included in there, and so therefore it's a highly flexible system.
So could you compare, for example, with using an LED disc screen, for example? You know, it's got a higher degree of pixelation, and will give you certainly more control as to the arrangement of the light patterns. So, you know, the complexity of assembling 96 LEDs versus buying a screen.
So I'm not quite sure if I understood you correctly. So of course we had several different designs in mind first time. So you could also use one high-powered LED and illuminate the 96-well plate from the top. But this way all the wells would be illuminated equally. And therefore we decided for this 96-well plate format in combination with the PCB. We can assure the optimal distance of each individual LED to the sample, and ensure a high energy transfer to the sample. And this design is done in a way that you can mount any LED onto this device with the same design, because you can adjust the current flow through each individual LED without changing anything in the design at all. So it's not only for our experiments, but for any light-induced experiments in the lab.
Okay, I think we run out from time of questions, but thank you so much.